非常感谢大家参加云端论脑第31期的活动。首先，我们还是要感谢脑人研对这个活动一如既往的支持。非常开心，今天请到了金鑫博士。然后金鑫本科在 MIT，然后博士的时候是在洛克菲勒大学，是从 Corey Bachman 是在神经科领域领军级人物之一，就是他是做线虫的一些环路行为。还有一些社会行为，反正是一个大牛之一。他还参与美国很多Arlotta，然后当然跟张锋有紧密的那个合作。张锋大家应该都已经知道，然后 Paolo 其实也是一颗冉冉升起的新星，就在我理解上，他应该是一个，就是他现在他也参与了很多，比如说美国的NIH 一些脑科学的项目，然后他也是其中一些他也是领导人之一。然后金星现在已经就是拿到了 Scripps Institute 的一个教职，然后他刚才跟我说他是7月7号实验室就开张了，然后他就说就国内有兴趣做博后，将来可以慢慢的再联系他。然后他有个做的非常好的网站，我觉得属于神经科学 Top 1的那种，大家有兴趣可以自己搜一搜，就是做的就是非常简洁，而且有美感。然后他今天讲座可能会用英文，因为他一直在美国这边找工作，所以主要是面对英语世界。然后但是大家提问，我觉得可以用中文，也可以用英文。然后提问呢，也可以 unmute， 就是可以取消你的静音，直接提问，或者你实在不好意思露脸或者说话，可以在 chat 里面提问，然后我会帮你就再向他来提问。然后那金星，我们要不先开始？好，谢谢。
好的呀，谢谢周丽的邀请，然后谢谢各位脑人员以及各位同事的帮助，然后我特别感谢大家做了一个很漂亮的海报，然后我这是第一次，其实在就是中文媒体的这个讲这个讲座，所以其实还有一点紧张。然后我很高兴能够在这里就隔着大洋。通过 Zoom 这个平台跟大家交流，然后像刚刚周丽介绍的，就是我现在这还在波士顿，在哈佛，然后我今天会讲的工作也是之前发表的在哈佛的工作，但是我下个月就会搬到 Scripps 然后呢，所以我就非常厚着脸皮的在这里给大家做一个小广告，就是如果在座的各位或者说各位认识各位对于就是包括 neuroscience 包括 tool development 这些方向感兴趣，可以来联系我。然后我们在 Scripps 在圣地亚哥，圣地亚哥呢不仅是一个这个做科学做biology 非常好的地方，它也是一个旅游胜地。然后能看到我们的这个 institute，就是能看到这个停车场啊和楼，就是在这个海边，所以你可以 set up 一个 PCR，然后出去冲浪，回来 PCR 还 run 着。我会非常鼓励大家来联系我，然后我们也刚刚做了一个网站，希望大家来参观指教。然后虽然这是一个sort of like a Zoom meeting，但是如果大家有问题，都可以 unmute，然后来问我。我希望能够用这个方式跟大家有一个 interaction，而不是一个单方向的一个 talk。好的，然后我可能会用英文来讲，因为就是 for the sake of convenience，but let me know if you have any question. So as I said, I've been really interested in developing tools, especially chemical biology and genomic tools to study the questions in neurodevelopment, as well as how these developmental problems sort of goes array in the case of neurological disorders. And particularly, we're very interested in developing a method to study gene function in a more scalable manner. And this motivation is really driven by this question when I think about neurological disorder, especially psychiatric disorders. Because in the past almost 2 decades, all these fantastic advancements in human genetics and population genetics, it has revealed that a lot of the disease and disorders actually have strong genetic roots, and especially psychiatric disorders. Now we know that in the case of autism spectrum disorder, for example, we already know a long list of genes that contribute to the manifestation of these disorders. So I'll start with a few examples. For example, one of the probably the most famous autism-related risk genes called MECP2. It is actually the mutation of which is solely the cause of a subtype of autism called the Rett syndrome. And in this case, MECP2 is actually encoding a protein that regulates transcription. And it can be a transcription activator as a repressor. And we now know through many people's fundamental work in the field that MECP2 is actually required for both the development as well as in the adult stage. But as I said, autism is not really a simple disorder, but when we shift our attention to a different sets of genes, for example, in this case, another gene that implicated in autism called SYNGAP. And in this case, SYNGAP is actually encode a GAP protein operate on synapses, basically the junction of neurons and regulating how neurotransmission. And now we know that they are part of glutamate receptor complexes and can regulate receptor trafficking. So you can see that SYNGAP already is localized in the different parts of the cell, a different compartmentalization, also regulate a very different biological process compared to the transcription modulator, MECP2, that I showed you earlier. And I'll give you one last example in the case of Fragile X, which is a subtype of autism. And in this case, the gene that's implicated in Fragile X syndrome called FMR1, it can regulate protein translation as well as epigenomic modulation. And again, a very different sets of biological process compared to the other two. So we can really go on and on about these single genes and monogenic models. And in the past, many people in the field has focused on these single genes and trying to understand the mechanism underlying. And in fact, these really foundational work, they have built important models to allow us to use many different methods, including electrophysiology, molecular biology, biochemistry, or even behavior models to approach this very complex question of autism. But compared to many other diseases or disorders, now we know that our understanding of neurological disorders are still at a very early phase. For example, for several of these genes that we just discussed, we simply don't know when and where do these risk genes act. And we also don't know how many different kinds of autism there are. So a few years ago, when we sort of started this project, our question was that among all these different autism risk genes and loci, how many different kinds of autism there are? And is it possible that each of these individual hundreds of risk genes actually represent an independent question and need to be studied independently? Or is it possible that subsets of these autism risk genes actually converge and diverge into a particular subtype and can be grouped together for mechanistic considerations? And I want to point it out that many of these risk genes that confer sort of risks in the psychiatric disorder are actually transcription factors and chromatin modifiers. So they do express broadly during development. So we wonder whether these kind of broad expression imply that they actually affect very diverse or maybe similar sort of convergent sites of cell types? Or is it possible that these risk genes even affect the same molecular pathways within these individual cell types? And these will be very important information to know in order for us to better model them and study the underlying mechanism. So as a start, we decided to focus on just a subset of these so-called autism risk genes. And these are what I would call the de novo variants, meaning that they are the kind of variants that's only present in the affected children, but not in the unaffected family members. And these type of de novo variants, that's often protein truncating and loss of function. And this type of variants, although they are very rare and only present in the trio studies, they are often highly penetrant and carry large effect sizes. So this is quite different from the kind of GWAS variants you might be thinking of that many other disease disorders approach are focusing on. But we decided to start with the de novo variant because of the larger effect sizes that make us think it might be an easier entry point to this very complex question of autism biology. But even when we decided to only focus on these de novo loss of function variants, we quickly realized that it doesn't simplify the problem at all. In fact, this gene list is still very long. And in the most recent and probably the biggest cohort analysis for these de novo variants that was published last year in 2020, the Satterstrom et al. Paper. In this list, they basically analyze almost 12,000 autism trios, meaning autism families. And from there, there's about almost 100 disease risk genes that's passing the statistical threshold. And you can see that on the y-axis on this plot is showing the statistical significance for each of the genes. And on the x-axis are basically all these risk genes. And you can see that they're distributing across all the human chromosomes. Some of these risk genes might be familiar to you, but some of these genes were not very well studied, at least when we started our project. And some of these risk genes back at that time didn't have the mouse genetic mutant model or even cell line model made yet. So, and as you can see that beyond these like big funds, famous autism risk genes, there are a lot of these risk genes sort of in between these bands with different kind of statistical threshold cutoff. And as you can imagine, this type of analysis is actually very sensitive for the data sets. And as we gaining more and more power to get these trios and sequence these trios, analyzing these data, you can imagine this list of autism risk genes are going to be growing bigger and bigger and longer. And estimably, there is probably hundreds and even thousands of genes that's contributing to autism risk. So now we know that this long list of risk genes is likely to get longer once technology gets better. So this really demands a systems genetic approach to study psychiatric disorders. So we propose this approach, and we hope that this will be a generalizable approach so that we can sort of start with a in vivo genetic perturbation approach to introduce the perturbation in utero. We will be able to do a pool in vivo CRISPR perturbation and only postnatally analyze these perturbed cells. And particularly, we want to use some kind of high resolution readout to ask what are the cell type specific effect that's associated with each individual perturbations. So the goal of this approach is that we want to be able to test the effect across many different perturbations. And we want to be able to assign their function in individual cell types. And there's a very important sort of assumption here, thinking about development, because we know many psychiatric disorders, especially autism and schizophrenia, they are usually diagnosed very early and they have a developmental origin. And therefore, it's very important to introduce perturbation in the context of a developing tissue instead of just performing this kind of experiment in the adult animal or studying the effect in vitro in the induced neurons. So in addition to the complexity of this long list of risk genes we have to go through, brain, as you all already familiar with, it's an organ that's really famous for its cellular heterogeneity, which of course presents another layer of the complexity of the problem. So in order to sort of study the gene function at scale, to study many genes at once, but also be able to assign their function into individual diverse cell types, we were very lucky and really stood on the shoulder of giants who developed in vitro Perturb-seq a few years ago. And our experiments are largely built on the published Perturb-seq system. So it works like the following. We can introduce mutations in the genes of interest, in this case, autism risk genes, through introducing the guide RNA targeting these genes. And we can inject the lentiviral vectors in the lateral ventricle of an embryo very early in development at embryonic day 12. And this is such an early stage of corticogenesis that we can imagine at that time, the cortex is not formed yet. And there are neuroepithelial cells, which are neuronal stem cells, sitting on the lining of the ventricle walls. And the subsets of these neuroepithelial cells will be labeled by the lentiviral vector, which confer the expression of the fluorophore, which is a blue fluorescent protein here, as well as the guide RNA, which allow the in vivo CRISPR perturbation for the given gene. So after the injection and the perturbation in the next 2 weeks, you can imagine through these in vivo and in utero development, all the cellular progeny that came out from these clones of perturbed cell will also carry the expression of the fluorophore, as well as the loss of function of the gene that the guide RNA is targeting. So 2 weeks later, when we are analyzing the postnatal day 7, you can imagine the cortex is a huge mosaic, sort of clones of perturbation cells. And we can basically use FACS to enrich these blue perturbed cell and use single cell RNA sequencing to read out each cell's perturbation states, as well as their transcriptomic profile. But single cell resolution in this stage is really required because it not only offers us a very high resolution phenotypic readout, but also allow us to multiplex this approach. So this means that we don't have to inject one vector to study one mutant, but we can actually pool all the different perturbations and do one injection, and only computationally to read out for each of the cells what their perturbation identity based on the barcode from the lentiviral vectors. And ultimately, through this type of experiments, our question are, what are the cell types and what are the molecular pathways that's most vulnerable through the perturbation of this cohort of autism risk genes? And I want to also highlight that in this particular case of experiment, because these autism risk mutation don't really co-occur in patients, we actually dilute our lentivirus so that most of the cells here receive one and only one perturbation, just because the genetic epistatic is not really the human genetic context information that we want to task here. So I also want to highlight that our approach obviously is a more efficient one because we can study many perturbation and many risk genes at once. But precisely because of that, our approach is more well controlled because we're always comparing the perturbation with an internal negative control that was injected in the same brain, that was dissociated in the same dish, that was FACS in the same population, that was sequenced in the same library. So this will allow us to actually build a linear regression model to regress against other confounds, such as the differences of developmental timing or sequencing batch effect, and therefore read out a more robust effect for each of the perturbations. So this particular experiment that we did that got published also comes with a set of features, such as what we're analyzing is actually a mixture of homozygous and heterozygous loss of function, because the sort of monoallelic perturbation is hard to control from this type of in vivo CRISPR experiments. We did tested our guides and screened our guides ahead of time so that we know each of the guide RNA will lead to effectively 40 to 70% of the frame shift mutation. And we include 2 guides RNA for each of the risk genes. And in addition, we also want to know that these clones of perturbed cells are actually sitting in the sea of wild type normal developing brain cells. And therefore, this type of context actually allow us to focus on reading out their perturbation associated cell intrinsic effect. And of course, that's readable by the RNA profiling. And last but not the least, we, in this case, introduced the perturbation at embryonic day 12 and analyzed the effect at postnatal day 7. So postnatal day 7 is a time that most of the cell types in cortex are born, but this is no way an endpoint of neurodevelopment. But in the future, it will be really interesting to perform the readout at a later developmental stages to focus on other neurodevelopmental events, such as naptogenesis. And you can also imagine that this approach can be applied to other systems and other model organisms as well, not limited to just mouse, and also can be used to study gene function in other tissue type as well. Okay. So in total, using this approach, we analyzed the effect of 35 different autism risk genes through the collection of almost 46,000 cells through 17 independent experimental batches. So before going to the single cell analysis, I just want to show you that our approach can be generalized to study many different cell types in even many different brain regions to ask how these genes affect neurodevelopment. We noticed that just 24 hours after injection of the lentivirus vector, we can already observe the fluorophore start expressing, meaning that lentivirus is a pretty quick virus compared to AAV or other kind of variants. And the timing is important for us because if the vector expression delay for 2 days, we're losing an entire population of the deep layer projection neurons. And at postnatal day 7, you may be able to appreciate that in the cortex, there's this very sparse labeling of these red perturbed cells that's pseudo-colored here. But we can also appreciate that there are many different brain regions that are also targeted by this kind of early injection of lentivirus. And that include hippocampus, striatum, thalamus, and so on. So in the future, it will be really fantastic to perform this kind of similar perturbation, but be able to analyze their risk gene effect in different tissues and really understand how different tissue contexts and cellular environment actually introduce these differences in terms of the risk gene phenotype. Okay, so from this type of single cell analysis, we collected about 46,000 of the single cell RNA sequencing profile, and we can classify them into 5 cardinal cell types, including neuron as well as glial cells. And these cell type can be pretty robustly annotated through the known marker gene expression, which I'm showing you here on the right. But I want to highlight that using Perturb-seq, what we are reading out is not just basically the classification of these 5 major cell type, but rather we can take each of these 5 cardinal cell type and further classify them into subtype. And for example, for the excitatory neurons, we can further classify them into subtype that correspond to their laminar positions in the cortex. And in this case, our readout of this assay is actually truly single cell resolution, because if the perturbation or the genetic knockout actually affect even 1 out of these 5 cardinal cell type on the left, or even just 1 out of these numerous subtype of cells here on the right, it will still be captured and analyzed in our pipeline and wouldn't have been masked like in the bulk analysis. So when we're talking about single cell analysis, you might be wondering how many cells are enough, and especially in the case of the Perturb-seq analysis, how many cells are enough for the phenotype that I care about? It obviously depends on many things, but most importantly, it depends on the type of analytical framework that you choose. And traditionally, we like to think about analysis like the one showing on the left, which is a gene level analysis, assuming 20,000 genes are actually independent variables. But we know that biology do not operate on black boxes and randomness. Biology actually operate on the gene networks. And if the perturbation indeed introduce any effect, we actually expect a network of gene expression to change instead of just 1 or 2 gene expression changes. So here, what I will show you is a downsample analysis from the published Perturb-seq work, just to show why we prefer this type of gene program or gene module approach. So this is actually analysis of 4 data sets of single cell of about 60,000 dendritic cells upon the perturbation. And this is represented at the top right corner. And then from there, we can actually start performing downsampling, either by moving on the x-axis by reducing the number of transcript, which is the sequencing depth, or we can go down on the y-axis, which is reducing the number of cells included in analysis. And you can see that if I move from the top right corner down, the correlation compared to the 4 data set on the top right corner dropped pretty significantly. That is because this analysis that relying on gene level single gene approach is actually very sensitive to how deep the sequencing is and how many cells are included. However, if I perform the similar downsample analysis, but in this case, relying on the gene modules, you can see that we can take the top right corner, go down pretty significantly, still maintain very high correlation compared to the 4 data sets. And this is consistent with our data sets as well as the published Perturb-seq data sets that about 30 to 50 cells per perturbation per cell type, we can acquire pretty robust readout. So in addition to gene module approach being more robust, just statistically speaking, I myself also like this kind of approach because I think it's more interpretable. So instead of 1 or 2 differential gene expression significant DE genes that you have to Google a lot or PubMed a lot and try to guess what is the underlying mechanism and underlying biological changes, in this case, what I will have is a list of genes that's already inherently showing as a pattern from our data, and is more likely to yield a more meaningful hypothesis for me to test. So the question is, how do we do this to extract the gene modules? We tried many different approaches, including testing existing geo terms, other type of module that was defined from independent data sets. But ultimately, what we found most fruitful is actually to learn de novo from our own data and learning the inherent pattern of our own data. So what we did is to take one out of these 5 cardinal cell type and one at a time, to basically look for the covariant gene modules across all the cells, regardless of their perturbation identity. So we're looking for genes that seems to go up and down together across all the cells. And from there, for each of these individual gene modules, we can basically identify what's their expression pattern is like, what's their biological meaning. And ultimately, we can set up a regression model to ask how are each of these perturbations affect the gene module expression compared to the negative controls. So I will show you what kind of module we'll be able to extract it from this type of analytical framework. So we used both the more traditional WCGNA, the weighted covariant gene network analysis, as well as a machine learning based topic modeling approach. And both method reveals quite similar covariant gene module, which makes us think that we're capturing the real signals from data sets. And for example, in the case of excitatory projection neurons, there are times we extracted the gene module that seems to be highly expressed in the particular subsets of the subtype of projection neurons. And in this case, this projection neuron 1 module, PN1, the expression of these marker genes are highly enriched in this sort of 4 o'clock, 5 o'clock population, which correspond to layer 4 projection neurons. And aficionado in the audience can recognize many of these genes' name and realize that they are very specific to this population of neurons. So we call these our subtype specific modules. But there are times we extracted a module that look like the one on the right, which is they're expressing across many different modules on these t-SNE plot. And in this case, the projection neuron 2 module, PN2, actually describe a biological process. If we look at the name of the genes, they are actually a module network that's describing neurite development. And neurite development is a feature that is conserved across all these type of different population of projection neurons. Therefore, you can see their expression across all the subpopulations on the feature plot. So we call these type of module a feature specific rather than a subtype specific modules. So we do include both type of gene modules in our analysis. So finally, we extracted a total of 14 different modules coming out from our 5 different cardinal cell types, which is shown here on the y-axis by row. And on the x-axis, each column represents 1 out of the 35 autism risk gene perturbations. And here on the dot plot, the color of the dot represents increase or decrease of the expression of the module, and the size of the dots represents statistical significance. So without going to the detail yet, you can already appreciate from this sort of 10,000 feet bird eye view that even in this 35 risk gene perturbation, we can observe certain level of recurrent phenotypes and convergence. So some of these effects are very specific to a cell type, such as the perturbation of this gene ADMP effect specifically in projection neuron. But some of these perturbations actually hit multiple modules and multiple cell types, such as the perturbation of this gene SCN2A affect both projection neuron and astrocytes, at least at this time of development. So this again highlights the power of the single cell analysis that can capture these phenotypes and also assign these subtle molecular changes into a particular cell type that they belong to. And as a neuroscientist, to me, one of the most surprising things is actually we observe that both neuron as well as glial cells are directly impacted by several of these risk gene perturbations. And this is really important information to know, especially in the future, if we want to model and study many of these risk genes in vitro, when the cellular heterogeneity is more limited. And it's important that to know for each of these risk genes, how to model them. Do we differentiate them into neuron or astrocytes based on what the cell type they actually impact? So in the in vivo context, we get to see everything, and having this comprehensive picture will guide us to better adjust our focus. So this is really a very rich data set that there are a lot of things to follow up on, a lot of potential follow-ups, but you might wonder, just like any genetic screens, what are these hits really means and what have we learned from these results? So I'll go through several of the key phenotypes before diving into a detailed set of validation experiments at the end. So what have we learned? We learned that these cohort of autism risk genes affect not just 1 or 2, but rather at least 4 different cell types. And these are projection neurons, interneurons, astrocytes, as well as oligodendrocytes. So this is also actually correlated with our co-analysis of our data with the human data, which I don't have time to show you now. But basically, we took the recently published paper, is a landmark work from Arnold Christian's lab that analyzed the human post-mortem autism brain samples using single-cell sequencing. And from these co-analysis, we also observed that these are the 4 cell types that show significant differential gene expression signature. And many of these strongly differentially expressed genes show similar change of gene expression and change of direction, also from the mouse in vivo Perturb-seq data. So these are definitely very intriguing correlation that make us to think about a lot of these shared developmental events or even autism risk gene related contributions across both tissues in mouse and human. I'll discuss this in probably 10 slides also at the end of my talk. But going into these detailed sets of phenotypes, first we noticed that in the projection neurons, layer 4 projection neurons seems to be particularly vulnerable at this time of development. And it's interesting because this is also the time that the thalamus, which represents sensory input, are being wired into this layer of cortex. So this abnormality that we're observing from layer 4 may be linked with the abnormalities in the sensory processes in autism patients, such as their hypersensitivity that many have studied with the recently the beautiful paper coming out from David Ginty and Laura Orefici's lab. In addition to the projection neuron, we also observed that in the interneurons, the perturbation of this risk gene ANK2 leads to an upregulation of a synaptic module, and specifically in this subtype of interneuron that express a neuropeptide NDNF. And in this case, there are several synaptic gene signatures, such as the potassium channel and GABA receptor that were upregulated. So we suspect that this is related to anchoring's role in regulating neural connectivity. And previously, ANC2 has been shown to have this role in the excitatory neurons, but this is its new additional role in interneuron, although in the similar pathways. So in addition to the neurons, we also observe that in the astrocyte, there are two different homeostatic gene signature in the protoplasmic astrocytes that are affected. But in the rest of the talk, I will tell you one example of this phenotype that we validated using a germline mutant N. And this is about a glia phenotype in the cell type called oligodendrocytes. So oligodendrocytes is a really interesting cell type that they can deposit myelin on the axons of neurons to modulate conductivity, among many other things. And the maturation of oligodendrocyte really take several months, even in the context of mouse brain development. And in our case, at postnatal day 7, in such an early stage, most of the oligodendrocytes in our data is actually progenitors, shown here in purple and red. They are oligodendrocyte progenitors, OPC. And there's also just a very small population of oligodendrocytes, shown here in cyan color, that is actually the newly formed oligodendrocytes. So we extracted a module called ODC1, and we found out that this module is actually not evenly expressed across all the oligodendrocyte cells. In fact, they are actually expressing higher in the top parts of the t-SNE plot, which is the progenitor population, and they're expressing lower in the bottom part at 6 o'clock. So you can think about this module are very enriched in the progenitors. They represent immatureness or progenitors. And when we examine this module ODC1 and see how different autism genes perturb the ODC1 module expression, we notice that the perturbation of two genes, one of them is CHDA, actually significantly lead to the decreased expression of this progenitor module in oligodendrocytes. So when we just saw these results, there was a paper studying CHDA in oligodendrocyte that just came out, and they used TRIP-seq to show that oligodendrocyte as a chromatin modifier can directly bind key genes that's crucial for the survival and differentiation of oligodendrocyte, which really gave us more confidence about our result assigning the function of CHDA in oligodendrocyte. So we went on and used the heterozygous mouse mutant line to validate our results. And first, I just want to directly validate what we have observed in the postnatal day 7 cortex. And we chose in situ hybridization to directly quantify these genes that we identified from the Perturb-seq approach, being the oligodendrocyte immature marker or progenitor marker. So when we quantify these marker gene expression, one of them being PDGFR, showing here, we observed about 30% reduction in the mutant, which is, again, the similar effect sizes that we observed in the Perturb-seq. So that's really assuring, but we suspect that this kind of like changing of immature signature is actually representing some sort of changes in the maturation timing or the developmental progress. So what we want to do is kind of a converse experiment, which is instead of examining the progenitor and immature marker, we want to take a matured marker. In this case, it's myelin basic protein. And in addition to analyzing RNA throughout this entire project, sometimes I do wonder whether there's any differences at the protein level at all. So we did a very simple immunohistochemistry using antibody against the myelin basic protein. We examine the level of this myelin basic protein at several time points. And showing here is at postnatal day 11, when we observed the upregulation of myelin in the cortex, suggesting the model of an advanced differentiation of oligodendrocytes. So because oligodendrocytes, this is really a relatively rare cell type at this time of the development. Without the single cell data, we would not know how to focus on this cell type, examining their effect upon the perturbation. So now we know that maturation and differentiation is affected in oligodendrocytes, but it really opened up more windows and more things to learn, such as how is the developmental difference of this glia cell can directly lead to the physiological or even behavioral phenotypes at a functional level. Because both at the level of cellular differentiation, as well as their final myelination of the oligodendrocytes, these processes are highly plastic and they depend on neural activity, environment, as well as the experiences. So this really leads to a very interesting idea that these glial cells can use the non-cell autonomous effect to modulate the neural conductivity as well as the circuit refinement. And these modulation might be in the very subtle ways. And in the future, we will look into whether this kind of oligodendrocyte phenotype persists into the adult stage, or whether this actually affected their roles in myelination deposited in specific brain regions, or their myelination onto specific neural subtypes. So I will spend maybe 2 slides to talk about an ongoing effort to continue developing these functional genomic approaches and use that to study neural development. So there are maybe 2 things I didn't quite tell you about. The first I alluded to is the human and mouse differences. Humans obviously have more neurons compared to mice. And more importantly, they have a much expanded upper layer cortex, recently expanded in evolution. And there are several of these human-specific or primate-specific neurodevelopmental events that have only recently been studied or appreciated and have not been studied extensively. And many of these human and primate-specific neurodevelopmental events or cell type could be relevant to understanding of these human disorders. After all these autism and schizophrenia, they are human-specific disorders. So we should keep that in mind. And several of these aspects of the human brain developmental processes can be studied in a very simplified system called the cerebral organoids, which Paola's lab has expertise on. And in addition to that, one thing that I didn't quite tell you earlier is that we took the cortex and dissociated to use FACS to enrich these cells for single cell analysis. But by dissociating that, what we're completely losing is the spatial information of these cells. We simply don't know where these cells are in the tissue. We also don't know where their cellular neighbors are or where their cellular environments are. So it'd be really great to capture these transcriptomic changes and information in addition, and also getting to know their spatial coordinates. So this is a recent project that with Paola's lab, I set up the Perturb-seq and performing these perturbation experiment in the context of the human cerebral organoid system. And in particular, we're interested in several of these progenitor cell population that's unique to human and primates called the outer radial glias. So we can perform the similar perturbation of candidate genes, just like what I showed you earlier using the lentiviral transduction. But in addition to the single cell sequencing, we can also use in situ sequencing to acquire both the transcriptomes as well as the location of the cells. So for example, in this very simplified paradigm shown you here in the middle in this cartoon, there are 2 orange cells that can be very similar in terms of their transcriptome, and therefore they're classified into the same cell type, but they might be sitting in vastly different cellular environments, and these are the information that we're hoping to capture. So through this really fantastic collaboration with Xiao Wang's group at Abroad, as well as with Aviv Regev, we are performing StarMap, which is a spatial transcriptomic methods to capture these transcripts, but in their nascent form and in situ. And this method will allow us to ask how each of these perturbations can alter the gene program, just like what I showed you earlier in the Perturb-seq analysis. But in addition, we also ask how are these perturbations actually affect the cell's ability to migrate to a particular location, or whether the nearby cells are actually impacted by these perturbed cells, or vice versa, how are the cellular environment can in turn to shape the manifestation of the phenotype in the perturbed cell. So I hope I'll be able to come back to tell you more about this project just in a few months. So I just want to summarize what I've told you today, which is that I hope to convince you that we built this experimental approach and hopefully a generalizable approach to study gene function in the context of psychiatric disorders. And I said this is a generalizable approach because this method is easily scalable and also modular. For example, in addition to just using the single cell RNA sequencing as a readout, I already showed you that we can use spatial transcriptomics in the future, also coupled with the single cell multi-omics readout to capture these high dimensional data to further refine what are the alteration and function across this panel of different risk genes. And in addition, these kind of genetic perturbation part can also be modular. So instead of performing these insertion deletion events in the genome, we can change this into CRISPR-A or CRISPR-I to subtly changing gene expression, or we can introducing a library of transcription factors of your interest to overexpress certain gene of interest or cDNA of interest to study their effect. And these can be done in the context of, and then vivo brain development. We also have collaborators using this to look at the skin development or different systems. So it's not limited to neuroscience or only to the brain development. And using the single cell sequencing approach, it really revealed really rich data sets and many insights, including that how neuron and glial cell type can directly be impacted. And these are important information to know in terms of the cell type specificity for individual risk genes and important for future in vitro stem cell-based modeling, especially. And in addition, we took several of these hits and performed the functional follow-up. And in the case of oligodendrocyte advanced differentiation, we hypothesized that this kind of risk gene perturbation may have its non-cell autonomous impact to influence the natural maturation in the brain. So starting from a pool screen in vivo, we identified several groups of rare cell types that may have been overlooked using the bulk approaches. We also validated several of these results, and some of them are done in the germline mutant animal. So I think this is really a very exciting time that these risk gene and loci, they are not only important for clinical reasons, but they're also key entry point to understand very interesting biology. Because these gene and molecule, they don't give rise to thoughts and behavior directly because they have to impact the nervous system to do so. And functional genomic approaches such as Perturb-seq really allow us to take this list of genes and molecule and ask what kind of subtype or subsets of nervous system do they act on or operate on to allow these function as well as these functions. So in the future, in my own lab, in case you're interested, will continue this theme of developing neurogenomic approaches and study the context of neurodevelopment as well as related developmental disorders. And we must think about these processes in addition to these genetic programs that every day there's a population of transcription factor expressing and new cells being born. These processes are also highly plastic and modulated by an array of environment and physiological cues. And sometimes these physiological cues even shape the phenotypic outcome so defined by the genetics. But the combination of gene and environment can affect very specific cell type and very specific brain regions to perform these kind of tasks. And I'm interested in development because I think this is a very interesting time in life that these early changes can have these effects during the critical period to have long-lasting effect. And if you think about these genetic mutations or physiological hormonal cues, for example, they are all very global factors. And in theory, they should have access to every single cells and every single molecule in the body, but they do act with certain type of specificity that our data has shown as well as many others. And to be able to reveal and identify these type of specificity is really the motivation of many of my research, because understanding the specificity of these global signals is important for us to understand the sites of actions of genetic as well as physiological impacts and how these brain cells are made and mature during this critical period itself represents not only a fundamental biology question, but also has strong clinical implications.
So in the future, in the lab, we will keep building the in vivo Perturb-seq approach. There are new technology development aspects to adding more modalities. There are new approaches to introduce perturbation to allow more scalable capture of these data to be able to perform a true genome-wide-like screen using single-cell RNA sequencing as a high-resolution readout. But in addition, there are two new directions that's growing in the lab, and one of them shown here in the left bottom is to adding a spatial transcriptomic readout. And instead of just doing this in the two-dimensional thin sections, we're hoping to develop a technology to acquire spatial transcriptomics in the true whole mount brain, that we don't have to section the brain, but rather acquire these complex genomic information volumetrically from the whole brain. And in addition, while working on autism and related developmental disorders, one thing that really fascinates me is that how these different developmental disorders are heavily shaped by physiological cues, such as the sex differences. And in the case of autism, it affects about 3 to 10 times more men than women. And the women who are affected by autism actually carry way more mutation load than men, which suggests that there's a female-specific resilient mechanism. And the molecular mechanism and cellular mechanism of these female-specific protection is now well established. There are a lot of these early hormonal theories describing these during early pregnancy, as well as the perinatal stages. There are male-specific surges of hormones that's basically absent in the female. And understanding how these very conservative and evolutionary powerful hormonal molecule and how can they get access to modulate early perinatal brain development, and what are these sex differences in early brain development that might be linked with autism risk gene perturbation is one of the other directions in the lab. And as you can imagine, autism is not a lethal disease, and it is important to use high resolution readout such as single cell analysis and even including spatial resolution, would be able to allow us to really identify and tease apart what are the subtle changes in the brain that's linked with the disorder stage, and whether we can use these approaches and technology to identify which is the secret of this female protective effect, and thinking about using this way to develop next generation therapies and novel drug targets. And with that, I'd like to thank my mentors, Paola, Feng, and key collaborator, Aviv. Without them, I simply won't be here. I work with very closely with a computational biologist, Sean, who's basically brilliant, and we did a lot of analysis together. I was very fortunate to work with two brilliant young scientists, Amy and Michelle, and both of them are already in their independent next stage of their career. And we are very grateful to have the support from a team of fantastic human geneticists that are Elise, Ben, and Mark, who shared the gene list before it's published, and also continue teaching us to think about human genetics. And last but not least, I will leave these slides on that we are going to start in the sunny California and San Diego. Our lab is very close proximity to the ocean, and it's a really fun city surrounded by really just fantastic colleagues in both areas of neuroscience, developmental biology, but also chemical and genomic tool development. So if any of these directions or the ongoing work or published work sounds interesting to you, please feel free to read more about us on our website or write me an email. Thank you for coming, and I'll be happy to take any questions.
非常感谢做了非常清晰的那个讲解，然后要不我先问一些问题，就是关于就是你那个 Perturb-seq 就是发现了一些 convergent 和 divergent 的现象，然后就是我想了解你有没有发现非常明显的哪一个 convergent 的现象，就比如说是不是同一类细胞类型的哪一个生理的通路发生了变化？
对，就是其实我觉得，
有一件事情就我们觉得挺有意思的，就是说很多的 effect 我们看到都很 subtle，我们并没有看到 general population 的 percentage change。就比如说，我们并没有发现，比如 oligodendrocyte 就不见了 ，interneuron 就变多了。There's nothing as striking as that, which kind of makes sense。因为我觉得如果是这样的话，大家早就发现了，对不对？但是得有很多的这种 subtle 的这个 signature change，所以我觉得 ultimately，就是往今后的方向来想的话，我觉得是很重要的一点，就是一定要用这种比较high resolution 的一些方法来 read 到，因为这并不是一个lethal phenotype 或者是一个非常非常强的一个 phenotype， 它更像是一个 functional differences。那我觉得 cell type level 的话，我觉得很有意思的就是说，我们看到了很多的 cell type 其实都有 effect。很多的 cell type 在 astrocyte 和 projection neuron 有非常就是基本上好像是 linked 的一个 effect。那这个其实我觉得很有意思，从 development 的角度来讲，它其实就是一个有可能是一个 progenitor population 有一些 aberrant sort of behavior in the differentiation， 所以他们的 impact 就是从project neuron 一直 link 到 astrocyte， 所以这个跟之前很多的这些hypothesis 也是非常非常就是吻合的，就是说包括有很多的这些differentiation complexes， 包括这些 helicase complexes， 所以有可能一些 hypothesis 我们现在 ongoing 在实验室在测试的一些想法，就是有可能是一个 developmental 的 timing differences， 就有可能某一个 cell type 的时间，但是一个 relative term， right？ 有可能他们的时间或者他们 maturation 的 timing 这个有一点不吻合。那其实 it's really interesting to think about 不同的 cell type 他们其实需要 coordinate 的，就是说当 synaptogenesis 开始的时候 astrocyte 需要does its job, right? Now, what if astrocyte does not know is the time that oligodendrocyte is maturing, right? 所以我觉得这是一个很有意思的事情，就是说 how does one cell type know the other cell type is doing something else? And when this kind of mismatch happen, what you will have is ultimately an imbalance or abnormality in synaptogenesis. 但它一定是很 subtle 的，对吧？那 ultimately that's what autism really is. That's how I think about it.
对，就是你提的那点非常对。就比如说新闻性和 EI balance 就是很重要的概念，所以两类细胞可能要有一定的协同的发育。然后 chat 里面有个提问是说 ，Can we use single cell RNA-seq to find more de novo gene mutation？ single cell RNA-seq 能不能发现 de novo mutation？
对，就是其实 de novo mutation 现在大家都还是在用这个 genome sequencing， 就是 DNA sequencing 在看，大家不会看 single cell RNA。然后我觉得 single cell RNA sequencing， 因为它非常短，它也不会 cover 这个比较长的这个 genome， 所以说如果是想就是从一个 gene discovery 的角度来看的话，我觉得大家还是多数在看 genome 的这个 DNA 的 sequencing。对，
是。然后还有一点技术方面，我比较感兴趣的就是你的文章里用的是一种 sparse labeling 的方法，你就可以保证，比如说感染的都只能只有一个barcode，但是假设你稍微再 denser 一些，你有可能会找到有2个突变，它可能表现就会完全不一样，这样信息量就会更大。然后你的 GLP 细胞也会非常多，做起来也比较轻松，但是唯一的缺点就是 cell autonomous 和 non-autonomous 的效应可能会混到一起。
对，我觉得这是一个 trade off， 就是说你愿意一次多收一些细胞呢，还是你愿意多做几个 injection， 对吧？所以说我觉得这是一个 titratable 的 problem。那我觉得一开始其实 in vitro 的 Perturb-seq 大家其实非常感兴趣的就是能够看这个 gene-gene interaction， 对吧？然后这也是一个很有意思的问题，就是说如果说有很多的 common variants，which individually 它们 effect size 可能都很小，但是有可能你有3个 common variants 就能够有个 phenotype， 但是2个 common variant 就没有。像这样的问题，我觉得是 Perturb-seq 非常适合来看的。这也就是就是 in vitro Perturb-seq 的 paper， 当时其实他们的一个卖点就是他们用 transcription factor perturbation， 但是他们其实是用非常高的这个multiplexity of virus 然后来看的。对。那我觉得 in vivo 来做，你刚刚说到那点就很好，就是说我觉得其实是一个 practical problem， 就你愿意收多少细胞，然后你有多少的 statistical power， 但是这些都不是 theoretical limit， 对吧？就是说如果说你能够做好一个 power analysis， 大概知道我 我有这样的一个 library， 那 library 肯定不能 genome-wide， 可能我觉得5个到10个要让他们 randomly 的就是 recombine， 就是in combination， 然后有一个非常 clear in mind what signal to look for， 我觉得这会是一个很好的利用。
对，然后还有一个就是因为我这段时间也在用一些库，你自己比如说构建你这30个 mutation 的库要花多长时间？
那看就是怎么样构建。那我们当时就是还是比较 conservative， 我们想要一个 viral vector 能够多表达几个 guide RNA， 所以这个构建起来就比较痛苦。就是我们其实是有2步构建，就第一步构建就是我们有2个不同的 vector， 把 guide RNA1、guide RNA2 都放进去，然后呢，我们把那个片段 PCR 出来，然后跟那个 final vector 做一个 Gibson assembly， 所以其实是蛮多步的，至少。对，就是当然就是这些东西你们都可以就是 multi channel 都可以做，但是这个还是一个比较手动构建，要这样构建一个 genome-wide 那是没办法，这不是一个 pool cloning。对，但是如果你是构建一个就是可能小于200的 guide RNA pool， 我都会建议你手动构建，就不要pool在一起，因为pool在一起就是你就没有分开，那你之后如果想就抽出其中3、4个重新做，那你还得重新做，因为它全都 mix 在一块了。所以我一般的 idea 就是，我觉得如果是200个以下，我都宁愿手做，然后200个以上，我会肯定把它们就是在 oligo 的这个成分就破在一起。对，然后那个时候做就比较就 technical 就比较小心，就包括 transformation 什么的都需要它比较 evenly distributed。
对，你说这点我还没想到，因为我自己其实没有构建过，我都是跟别人借别人的库。的确，你用别人铺在一起的库，你将来想用其中单个的某一个，你还是要重新去找。
没错，所以就觉得说所以就是一个 bell curve， 就是说到哪里那个就是划不来了。但对我来讲，那个就大概100个、200个，我都还愿意手做，再往上我可能会铺来做。
明白。然后我们看有谁想直接提问的，可以直接取消你自己的静音，然后直接提问，或者可以在 chat 里面打出你自己的问题。对。所以你实验室其实将来的方向还是以发育为主，对吧？但是你会看一下后期的这些行为。
对，就是我觉得还是以这种就是发展技术啊，然后用一些就是新的一些这些 genomics 的方法，去看这个，主要还是看就是 perinatal development， 可能不会看得特别早，还是看到基本上 cell type 比较成熟之后，然后包括这些 hormonal signaling 都是大概 perinatal， 就是刚出生到 P7 之间的。然后我想知道为什么就是说这些changes transcription factor 啊，能不能够怎么样跟这个 autism risk gene perturbation 去互相影响？
明白。
晶晶，我可以问一个问题吗？
可以呀。
好，我叫吴清峰，对，在中科院遗传发育所做 PI， 反正也是中年 PI， 对，非常好的报告哈。对，我的 major concern 是关于这个 cell type composition perturbation 这个。因为我不确定，就是说你的每个 gene perturbation 是导致了这个 cell lineage 改变，还是只是你在 sort out cell 的时候，它本身的就 cell type composition 就不一样。比如说这个每个 perturbation 里面，你刚才说到有大概30到50个细胞，但这30到50个细胞可能非常 heterogeneous， 里面有 neurons、astrocytes、oligodendrocytes， 然后里面可能是 excitatory neurons、inhibitory neurons 都很不一样。像这种就是说你们是怎样去排除这种可能性，就是到底是 lineage 的改变，还是整个的它就是改变它的，就是只是这个 random 出现了这样的 cell type composition 导致的这样的一个 gene module change。
对，这是一个很好的问题。就是说我觉得很重要一点就是一定要足够的 repeat， 就是 single cell experiment， 如果你只做一次的话，虽然有1000个、2000个细胞，但它只是一个实验，对不对？所以说呢，我们其实是做同样的实验做了17次，然后17次作为 single cell experiment 是 unprecedentedly high in number，right？ 所以如果说一个 effect 是 significant 的话，我们是其实在 linear regression 的时候把这个 batch 加进去了，它必须
就是说 across 17次都非常 consistent， 这才会被我们 pick up。所以而这17次每一次里面它都是一个破，就是说你其实就是做同样的一个实验，把它 repeat 了17次。所以这一点我觉得是很多 single cell experiment 都需要重要的，就是说首先 single cell experiment inherently 就是 noisy， 因为你就是 hugely subsampling， dimensionality 很高。然后其次就像你说的有很多的 technical 的这些 differences， right？ 但是如果我们每一次 dissociate 的时候，所有的 perturbation 他们都经历了同样的facts， 同样的 dissociation， 同样的这些 technical steps。如果说是一个 random noise 的话，一般来讲是你会觉得它就是17次都出现 random noise 的这个概率是很低的，所以这是一个 statistical question。对，
好的，非常好。对，然后第二个问题就是你为什么会拿到那么多的 microglia cells？ 好像我们做很多 electroporation 的时候 ，microglia 看到相对来说比较少。
对，这是一个很好的问题，我们也看了很久，就是为什么里面会有 microglia。我们用 electroporation 的时候确实是看到的不多，但是这个我们用的是 lentivirus。我们不是很清楚 lentivirus 的 tropism。然后我可以comment2件事情，一个就是我们一开始看到这个 data 发现有很多的 microglia， 然后我们其实到了 single cell data 里面去看，然后这些 single cell data 的 microglia cell 里面呢，我们能够 directly detect 到这个 lentivirus 的 fluorophore 的 expression。因为我们当时想法就是说这些 microglia 他们因为首先我们要 facts 那 pick up， 对不对？那 microglia 有没有可能是它eat up 了别的 cell， 然后 indirectly 的 pick up 这个 lentiviral 的这个 expression。 所以我们想说如果这个是一个假设的话，那我 expect microglia 它的 endogenous 的这些 viral expression 会低一些，因为毕竟他们不是自己genome integration 然后开始就是 transgene express 的。但是我们并没有看到BFP 的 expression 在 microglia 里更低，它跟其他的 cell type 都一样的，因为我们用的是一个非常强的 promoter EF1 alpha， 然后我们都看到在 microglia 里这个 BFP 这个 fluorophore 是 top 5 的 highest expressing gene， 所以这个就是一个 evidence 说有可能这些microglia 不知道为什么是 directly pick up 这些 lentiviral 的。然后还有一个就是有可能我们在 injection 的时候 inject 的就是比较深，可能打到了就是 parenchyma 或者一些其他的 region。还有一个就是我们可能 introduce 一些 local 的 lesion， 有可能 recruit 了一些 microglia， 因为我们 inject 的时候是 E12，那个时候 microglia 已经它已经就是 cross 的从 yolk sac 已经 migrate 过来了，它已经在 deep layer sitting there， 所以其实还挺有意思的。然后我们其实跟 Beth Stevens 也谈过，然后因为他们实验室有人就是想要 engineer 一个 virus 专门来 target microglia， 所以我们还在用一些这样方法，看能不能做一个 in vivo 的一个 microglia 的一个 tropism screening。来想办法 enhance 这个 staining。 但是还有一点我想要说的就是说，还有一个就是说那个 percentage 不是 reflect 真正的在 brain 里的 percentage， 因为我们在就在 immunohistochemistry staining 的时候，我们能 detect 到 microglia， 但是并不是就是15%啊，这么是%什么。
对，因为你这个 whole cell single cell RNA-seq 在 P7 的时候，你 dissociate 会损失一些 neurons， 这个是没有办法。
对，因为我们要 dissociate， 而且我们还有 facts， 所以到最后其实那个血色散之物都跑来跑去，以后可能会用 nuclear。对，我觉得 projection neuron 是最 vulnerable 的这个时候。对，所以有一些那个ratio 并不是真正的它的 ratio， 但是我们觉得 labeling 应该是有一些 labeling， 但是可能 population 没有那么高。对
，OK。然后就 last question 就是关于就是autism 很多是这种 heterozygous 的 loss of function mutation。
对。
就是说你因为您刚才也提到这个在里面，你可能可以看到 heterozygous Perturb mutation， 也可以看到 homozygous Perturb mutation。你有没有比较过 control heterozygous 跟 homozygous 这种 perturbation 之间的差别？特别是后两者还是 homo。
对，这个其实挺有意思的。这个今后我们其实现在在写一个 grant， 就是想来就是 comprehensively 的看这个事情。我们其实有尝试过，就拿一些 cluster 让它能不能就是 further classify， 然后有一些问题就是那个 data 比较难 classify， 是因为
有些这些 gene 它并不是在这个 stage highly expressed，好多都是什么 chromatin modify， 可能几天就是在 embryonic development 的时候 express， 在这里并不 express。然后还有一个就是 single cell， 它比较 sparse， 如果你看不到这个 gene， 不能说这个 gene 就被 knock out 了，对不对？有可能是这个 gene 它就是没有被 capture 而已。所以我们尝试一些方法，说能不能 computationally 来 clean out， 说比如说这1000个 cell 多少个是homo het wild type， 我们没有得到太好的结果，所以我们没有 try super hard about it。但是我觉得in the future， 第一就是说，有2个做法，一个就是说，如果说比如说我们做 in situ sequencing 的时候，我们可以加一个这个 probe against this given gene， 我们就可以 quantify 说每个 cell 它到底是2倍、1倍还是没有，可以就是说收集这些 data 之后，然后把这些 data 都 classify 出来，然后来看到底有没有这个 allelic specific effect。然后还有一个就是说，其实可以想办法控制 CRISPR 的这个allelic 的这个 effect。就比如说，你可以找一个这个SNP 很多的一个老鼠的 background， 把它们 cross 起来，然后你知道这2个maternal and paternal allele 它们的 SNP， 然后你可以 design guide RNA 只 target 一个 allele， 不 target 另外一个 allele， 就是说保证你做的这个 CRISPR 是 100% monoallelic if anything。这个也是可以做成，但是我觉得 ultimately， 还有一个问题，就是因为这些 gene 其实都还就是说就是很多至少有一些 gene 被证明就是说是 required for development， 如果是 homozygous 很有可能就是 lethal， 对吧？但是这是我们做这个 sparse labeling 比较像 conditional knockout， 所以也不好说。但是 because we go through development and knockout， 所以很有可能其实 during facts even right 这些 knockout cell 有可能已经死了，就是 we don't know， we really don't know what we are getting。对，但是你说这个是一个很重要的问题，因为这个 gene dosage it's very important to think about autism。那我觉得screening 是一个方面，但是另一个方面，如果我们能够 narrow down 一个 cell type 之后，来 compare 不同的这个gene dosage， 肯定是很有用。
好的，非常感谢啊。
谢谢。I think there's a question on the chat. Are there any computational tools to find coding gene variants for 10x single-cell RNA-seq, or can use long-read single-cell seq to find them? The question is about coding gene variants. 一般来讲，就我 assume 这个问题是关于 human genetics。因为single cell sequencing 很贵 ，single cell 的任何 DNA 或 RNA sequencing 都很贵，所以如果你想要找的是一个 coding variants 的话，我觉得大家都不会用 single cell， 因为是一个 genome variants， 对吧？所以就 bulk sequencing 应该就可以了。那 bulk sequencing 的话，你说的很对，就是 long read 很重要，因为我前阵子还听到这个 Evan Eckler 给一个 talk， 然后他的意思就是说，所有做 genome sequencing 的人现在都重做，就用这个 long read 看到的东西和以前 short read 看到的东西完全不一样。就 we're talking about very basic human genome assembly， 甚至都不是所谓的这个disease control differences。所以 long reads 是非常重要，尤其是 in terms of genome coverage and genome sequencing, especially for these repeats and things like that。但是用 single cell 来看 human pathology data sets。这个还是非常少的，因为 disease spectrum 很大，然后this kind of experiments very expensive。Arnold 实验室有一篇就是 Vemoshev et al 这篇文章，然后最近有一篇 bioRxiv， 我记得是 Dan Geschwin 的，也是看好像也是 autism 还是 general spectrum of psychiatric disorder， 然后他们也是做了一些可能有大概20-30个 patient 的。20-30个 patient，20-30个 cohort 这种 single cell， 但是20-30个就是 it's already very expensive and difficult， 但是对于真正的这种 trio analysis， 我们讲的都是几万个做的，这个用 single cell 做还是比较 costly。
对，然后我觉得他的问题就是可能是想问那个 gene isoform， 比如说对，然后我们实验室其实也有这个方向的课题，然后的确有些算法可以从 10x sequencing 里面，不过非常不准，就像你说的 10x 测的非常短， 因为我们你把一些
是
短片段强行拼接起来，可能只是一个 artificial 的 isoform， 并不是它原来真实的 isoform。对
对 isoform，
嗯
Jeremy K 最近有一篇很有意思的文章，他一直在看，不就是这是 retina，不是在
嗯
这个 central nervous system，他看到了一些很有意思的就是 isoform specific 的东西，然后他也是对这 sequencing 非常 picky，我觉得这样的 isoform 这种 level 的这种细节，我觉得还是要 long read in general，这个 short reads 就是像 single cell 顶多 sequence 100个 base pair 了不起了，这个还是非常非常难的。
对，然后是有问题，大家可以直接按钮自己提问，也可以比如先在 chat 里面打上几个字，让我们知道就是有谁有问题，我就不会再不停的说话打断你的提问。我们先就是大概等个1分钟，因为这种 Zoom 的这种聊天，有时候就是这种情况，可能网速的原因啊，或者准备什么，跟面对面的聊天还是有差异。我们先这样先等一等。
没关系。
嗯。
对，我觉得 Zoom 最难的就是，要不然就是大家都不讲话，要不然就是大家一起讲话。这个
是的。你刚才提到你们也有在做那个 organoid 的 Perturb-seq 对吧？就是正在投的对吧？你们那个 organoid Perturb-seq。
对。
你好，我是廖庆龙，就是在贝勒医学院。我有一个问题，就是我有这个技术细节我有点不太懂啊。就是你做这个 Perturb 那么多基因，就是一个 cell 里面它只 Perturb 一个基因吗？还是说有可能会有多个基因被同时 Perturb？
对，这个就是看用什么样的 virus 以及 virus 的这个 titer 有多高。那我们用的是 lentivirus 本来就是一个比较差的一个 virus，这个 production yield 很低，所以我们就是染出来，我们可以看 immunohistochemistry，基本上就是说 perturbed cell 周围都是空荡荡的，没有 perturbed 的，它是一个比较 sparse 的一个 labeling。然后如果改用 AAV 或者是其他的 virus 的话，那肯定会很不一样。对，所以我们非常 intentionally 的把低度降低啊。还有一个就是我们 analysis 的时候，因为我们可以抓到就是说一个 cell 有几种不同的 barcode，那如果有一个 cell 有2个 barcode，有2个 perturbation，其实我们没有 analyze。对，所以我们就这次就是专门只分析了single perturbation。
就是说我这里因为问这个问题原因就是说，像很多自闭症啊这种 ASD 啊，它有一些并不是 single 基因的这个 disease， 它有一些是 multigenic 的 disease， 就是这个个体它其实是有好几个基因有就功能的异常，然后最终导致它这个疾病。就是你这个办法有没有什么样的办法能够就是说在这上面有所 contribute advance 这个 field， 让它就是说你可以得到更多的信息嘛？因为很少会有单个基因，就是说有一些像 MECP2 啊这种可能是比较明确的，是一个基因可能导致这个个体有明确的这种表现，但很多基因的话，它是多个基因都会有一些多多少少，但是呢，就是你说的那个 dosage 的问题，它这个有一些 dosage 的缺陷，然后另外一个也有 dosage， 然后 combine 在一起，最终导致这个个体有这个表型。所以我不知道你这个方法在这方面会不会有一些就是说应用或者怎么样的。
对啊，就是这就是为什么我们一开始选 gene list 的时候，其实我们选的就是你刚刚说的 monogenic models，我们选的都是 de novo variant gene。那 de novo variant 就是只有一个 gene，这些 mutation 在病人里从来没有被发现他们 co-localize， 就是没有一个病人是有2个这种 large effect sizes gene 的。然后你说的很对，就是说其实很多就是说这种所谓的 de novo variants that's contributing about like 15% of the patient cohort，很多的人就是85%的都不是这样的 de novo variants，他们有很多其他的原因。 那最常见的原因就所谓的这叫做 polygenic risk score，right？ 那这些我就觉得是一个很难解决的问题。I can even argue that at that level you can study it in mice, right？ 就是说，如果1-2个 loci matter， 那genome differences, right? Like structural differences. 所以我觉得那些的话，Paola 的实验室，包括 Kevin Egan、Steve McCarroll, everybody， 在一直想用这个就是 human-derived organoid vitro system to model that。但是那个是一件就是还是很 challenging 的事情，就是说，you know, first of all, it's vitro system， 然后cell line 虽然是 recapitulate human genome，but how many are we talking about？ 我们需要 grow 10万个 cell line 来看到这些 diversity 呢？还是因为这些都是 PRS prediction，they're not deterministic， 并不是说你可以说哦，我有5个 variants， 那我就会就是 manifest autism， 那4个就不会。There's no evidence so far in human genetics that you can absolutely predict. It's all population distribution。所以我觉得这是一个 holy grail。如果有人可以就是第一 classify， 第二understand 怎么样去做这些事情。但是 the promise is 就是一开始的 Perturb-seq 的文章，他们有一个很强的一个点，就是说它并不是说你做了一个 variant， 然后发现没有 phenotype， 那做第二个 variant 你要重新去做，它可以同时有好多的这些perturbation 让他们去 interact。然后我觉得如果 in the future， 这些如果说我们 narrow down， 比如说10个 variant， 我们觉得这10个 variant 可能中间一些 combination 会有一个 particular phenotype 的话，那我觉得你可以用一个非常高低度的 virus， 然后让这10个 variant 在vivo 或 vitro 或 whatever contact， 让他们这种自由的去组合去 perturb， 然后来找那个 phenotype。我觉得这会是一个很好的一个筛选的方法。但是我并不觉得 human genetics， 至少在 psychiatric disorder 这里 human genetics 到了那一步，就现在看起来这个 variance 还是没有办法被 narrow 到。
好的，谢谢。
你好，有一个问题是 from statistical point of view 来问你的。我看到一个 slide 是 9ASD region affect 5 gene module across 4 cell types， 里面提到个 adjusted p-value， 你是用什么样的 statistical model？
对啊，我们就是用了一个 linear regression， 然后 correct for multiple hypothesis correction。我们用了好几种不同的 statistical model， 我们在 paper 里 report 了一个，然后 supplement 里头也 report 一个，你可以看到他们是 highly correlated with each other， 只是一个比较 stringent， 一个比较 less stringent， 所以我们最后 report 了比较 stringent 的那个，但是general 就是这个 number 就是 I think 一个是我们 report 了9个 autism restraint 是 significant， 另外一个可能是12个 something like that。但是我觉得很多是it's hard to say 我们有没有 reach saturation， 就是说如果我们这个实验室做了17次，那如果我们做了50次it's gonna be 3 times more expensive， 但是我们能不能看到更多的 phenotype， 我觉得这个就比较难说，因为这是一个 genetic screen。我觉得像任何的 screen 都是你看到的你可以去explore， 你可以去 validate， 但是你没有看到的是因为 phenotype 真的不存在，还是因为你 underpower， 这个是很难说的。对，但是我觉得像我们刚刚说到的，就是今后这个 technology 有很多可以进步的空间，然后其中一点就是因为 sequencing 现在越来越便宜，然后single cell 的这个data quality 比起我当年做也越来越好了。如果让我们重新再做一次，就是说我们下一个 version 的话，我觉得 cell number 上还有 data quality 上都会有更有长足的进步。所以 statistical question I think it's more a threshold question， 就是 how confident you want to be。然后 ultimately 我觉得任何 screen validation 都是很重要的，对吧？就是说，就算是做这种普通的 CRISPR screen non-single cell CRISPR screen，你也是需要一个 independent allele去 validate， 然后包括ultimately 如果我们感兴趣一个 biological process， 我觉得 independent validation 然后找到 signal pathway 还是很重要的。
所以我就 assume 你这边可能用到 logistic regression to investigate correlation between outcome and covariate。
我们没有用 logistic， 我们用的是一个 linear regression， 就是我们发现 linear 就 sufficient。对对对，这个我想 original Perturb-seq 他们也是用一个 linear model。对对对。
OK，
对，我们基本上都是用 linear model， 因为这个不是一个 binary 的。
OK， 就像刚才前面那位亮先生提问的，就我的症状，那可能是 multi-gene control trait。那也就是说，在我看你的 graph 里面有好多基因，那就说可能其中好几个基因是就是 multi-gene control 同一个那个phenotype。像这种情况，你没有做一些，比如说像从统计来说，就 cluster analysis 可以找出 group of gene， 可能它们是 work together to control one phenotype。像这种也没做到这种。
Yeah, that's a very good question. Let me just show the slides again. 所以我想再重复一下，就是说我们看的 gene variant 是这种叫做 de novo variants。de novo variants 意思就是说这个 variant 在autism 的病人里有，但是他们的家人里都没有，所以它是 de novo。然后 de novo 是非常非常 rare 的，很多就是 I forgot, it's like I don't know, it's like the rate. The rate is very chance is very slim， 所以这些我们 study 的 gene 里都没有发现 combination， 就是说没有一个 patient 是同时有2个的，更不可能有2个以上，所以我们专门选了这种非常 de novo loss of function variants， 用这个来 study。但是你说的对的，就是说有很多其他的一些non-coding variants, SNP background changes and things like that， 他们肯定是 modulatory 的，但是这并不是我们 study 的这个对象。
OK， 然后就在一个 question， 就说您有讲到有几十个 cell 来做实验，就说你这个 sample size 多大，然后你从那个 p value 来看，你就说 sample size 是不是 sufficient for hypothesis testing？ 就 sample size 多大？
对。对，这里有一个 downsampling analysis， 对不对？就是说sample size 有多大 depends on what's your readout, right？ 如果你用 gene level readout 的话，我们这个 downsampling 从这个6万个 cell 开始 downsampling， 它很快就变成不 sufficient 了。所以it really depends on the statistical framework。对，就是说这个power analysis， 就是说 single cell 是要 depends on what are you looking for, right？ 就是说你是要找的是 rare cell type 的 representation 呢，还是说一个 particular phenotype 的一个statistics？ 所以我觉得这个 graph 就是一个很好的 summary， 就是说 gene module analysis 对于同一个 data set way more robust compared to gene level analysis。OK，
谢谢。最后一个 question 就是说你用那个老鼠做那个实验，那我觉得为什么不用 monkey 呢？就猴子从进化角度来说，会不会更接近人类？
我们没有 monkey 啊。
就是说它的 model 动物的模型的话 ，monkey 会不会更适合做比起老鼠来说，会不会更适合？
对，我同意但是就是 practice speaking， 我们就是没有 monkey， 然后我们也没有想过说有这个 resource 来做这个实验。但你说的对，就是说从 model similarity 来讲，肯定 non-human primate is probably better than mouse。但是我想 argue 的就是说，也许从 practical perspective we probably understand more about mouse brain。就说我们在做 single cell analysis 的时候，我们有更多的 ground truth， 但是对于 monkey 的话，因为我们了解的还很少，所以但是理论上来讲，就是说这个实验是可以在很多的 model organism 里面做的。就是说我们虽然在老鼠里做，但不代表这个只能在老鼠里做。那 in the future 如果的话，我很愿意在其他的 system 里做。
OK， 好，谢谢。
谢谢。
对，其实，在大动物物理做，其实挑战会更多，对吧？你的区域的选择以及而且在欧美国家做猴子非常艰难，很难做，所以中国本身在做很多猴子相关的东西。现在，
对，这个确实是很大的一个优势。对，
对，美国这边大动物感觉做猪的比较多，比如说 Elon Musk 也在猪上做了实验。
对。
看一下还有没有谁有问题，给大家30秒的时间。 然后就是你们是有问题，其实可以就是直接打断我，因为我主要是怕就是讲座太冷，大家这里觉得很尴尬。好，你问。
金鑫老师你好。
你好，你好。
我是山西大学的姚明泽，我想请教一个问题，就是我不知道您这个测单细胞在小鼠里面测这个单细胞的时候，它有没有做这个对照呀？就是是不是需要转一些 S 样的对照？在这个
对
您在刚开始注射的时候，这个对照的话 ，S 样对照的话转多少比较合适啊？
对，这个是很好的问题，对照很重要。这个主要还是看你有多少的 targeting gene， 就比如说，一般来讲，如果是在 CRISPR perturbation， 就是说我会想至少5%到10%的这个对照是最好的。然后对照有选要做什么样的对照，很多人用这个叫做 scramble RNA， 就是完全是 synthetic， 没有跟任何的这个基因里的那个序列是有关的，这是一种对照。但是这个对照的问题就是说，这个对照在细胞里其实那个 Cas9跟它这个结合了之后，它不会切任何东西。但是呢，你的真正的这些 targeting gene 呢，其实它的 genome 其实被切断，它有一个 DNA 双螺旋的这个断裂，对吧？那其实很多人也会想要做一些对照来就是找一些，就是你认为不会有功能的一些地方，然后让这个 Cas9 真的去切割，我们叫做 targeting control。这两种 control 我觉得都很重要，就是说做实验就是 control 越多越好，对照越多越好。
好的，谢谢。
不客气。
郭元猛，你有问题吗？
我那个不好意思，我的 video 好像start不了，然后它总是显示什么 host，好了，我想问一个就简单的问题，就是为什么选了12.5？
因为我想再往前我做不了。
但是因为它本身很多都是 development 的，这个跟人上的有更早的吗？我不太清楚这个小鼠这个发育时期跟人的到底怎么去 match。
就是这个是 practical 的问题，我没有办法在E11做就是没有任何的，就是就都死了的。对，那这些在人里头它都是 germline mutation， 就是他们在它不是一个 mosaic 的一个 mutation， 它是在就是最早的这个细胞里头都有。对，所以你说的对，就是说越早做越好。但是我没有办法，也有可能，我当时有想过，就是有人说用一个特别的一个手术灯什么的，可以做一些更早一些这种 neural injection， 因为它要 inject 到 lateral ventricle， 所以理论上只要 neural tube close 之后，应该就可以可以做，但是这个就越早就越难吧，因为就看不出来它那个形状。
还有一个比较小的问题，这个你这个同一个 neuron 应该不能被病毒感染2次，对不对？是不是这样？
有，但是非常少，因为我们病毒的这个量很少，所以它就是一个 Poisson distribution， 就基本上就是0和1之间。对。
如果你病毒的量很少，那就应该不会影响，比如说某一类的病毒对另外一类病毒的它的比例，感染这一类细胞的应该不会有影响。比如说就是这互相的这些菌，它不会有影响，它不会影响另外其他的菌的比例，就是说
你的意思就是说它们之间会互相影响？
对，它应该不会影响。如果你的病毒的比例非常低的话。
对，就是说，如果你病毒比例低的话，它就影响比较少，但是收起来也比较痛苦，因为就细胞比较少，所以对。 对，
谢谢。
没关系。
对，刚才金鑫也提到了，比如说 E12其实是皮层干细胞，就是刚开始产生神经元的时候，就 neuroepithelia 到那个就是皮层的那个干细胞的反应。那如果没有谁再提问题的话，我会先把这个视频录制先关了，然后再看有没有人感兴趣的，可以跟你聊一聊。然后比如说如果没有人的话，我们